Hello friends, today we are going to solve AS level structured question October-November 2023 and component code 23. So let us move on to the first question. So the first question states the malarial pathogen Plasmodium falciparum enters red blood cells after a person becomes infected. After some time, each cell of Plasmodium falciparum divides to form daughter cells, and figure 1.1 shows cell of Plasmodium falciparum that is forming many daughter cells, and from the figure it is clearly seen they have given the information about cell surface membrane of the red blood cells and nucleus which is present inside the uh, Plasmodium falciparum, the daughter cells of Plasmodium falciparum. And the question A, with reference to figure 1.1, suggests how the presence of Plasmodium falciparum affects the red blood cells. So here we are going to suggest how the presence of this organism is going to affect the red blood cells. So we all know that red blood cells are biconcave disc shaped. So the presence of this uh, organism, Plasmodium falciparum, changes the cell surface membrane so that the cell surface membrane of red blood cells become irregular. Then the Plasmodium falciparum daughter cell fills the red blood cells. Because of this, red blood cells will burst. Then what happens? Hemoglobin will be used up by the pathogen. Since they are inside the red blood cells, the hemoglobin will be used by the pathogen. Since all the hemoglobins, I mean most of the hemoglobins are used by the pathogen, the oxygen will be transported in very less quantity to other parts of the body. So wherever the blood is moving inside our body, only less amount of oxygen will be transported in those blood cells. Second part of this question B, when Plasmodium falciparum divides, there is unequal division of the cytoplasm to form small genetically identical daughter cells. Outline the events that occur in the cell of Plasmodium falciparum to form the daughter cells shown in figure 1.1. So when Plasmodium falciparum when they divide inside the red blood cells, they will not immediately form into a new daughter cell. Instead, they initially form a multinucleate stage, a structure with multinucleate stage, and slowly they divide into single daughter cell which can infect the other red blood cells. So during this process, during this event, what are all the events that is going to occur in the Plasmodium falciparum we are going to look. So first, the pathogen is going to synthesize its own biochemicals, then it's going to produce its own organelles, it is going to synthesize its own DNA, then it also produces many nuclei, then cytokinesis which forms the cleavage furrow. So this, after forming the multinucleate stage, cytokinesis will be formed, cytokinesis which takes place in the cleavage furrow, and finally the daughter cell will bud off. C part of this question, um, explain how antibodies will reduce the spread of malarial pathogen through bloodstream. So whatever the information given above the question is just an information of about what World Health Organization has asked some researchers to do for the producing vaccine for malaria and all those things. But the actual question what we want to look here is explain how antibodies will reduce the spread of the malarial pathogen through bloodstream. So we know that antibody will always bind to the antigen which is found on the surface of the pathogen. So once they get binded to the surface of the pathogen, how they are going to bind? Because antibodies can only bind to a specific antigen because they have their complementary shape to the antigen. Then this is going to prevent, once the antibody binds with the antigen, this is going to prevent the entry of pathogen inside the red blood cells or liver cells, as the malarial parasites, they can enter inside red blood cell or liver cell to cause damage and multiply. This will prevent the pathogen entering inside the red blood cells or liver cells. Then antibodies is going to mark the pathogen for phagocytes. This stimulates the phagocytosis. So as we all know, phagocytosis will come and engulf the phagocytes will come and engulf the pathogen. Yeah. Moving on to next question. D. Some vaccination programs have been more successful than others. discuss the factors that contribute to the success of a vaccination program. So here we are just going to explain about what are all the factors that can contribute to the success of a vaccination program. So it is a general question where we can talk about the availability of vaccine. So vaccine should be available for large group of population so that the herd immunity can be achieved. Then vaccination should be produced in such a way that it should be more stable so that it can be stored for long period of time, and they have uh, long shelf life, and they can also withstand high temperature. Then more people trained to deliver or inject vaccine. So creating more vaccine or producing more vaccine is important, and moreover we should also train more people to inject vaccine so that more number of common people will be delivered with the vaccine. Then creating awareness to people regarding the benefit of vaccine is also very very important. Only when the proper awareness is there, people will come forward to take up the vaccines. Then proper government funding to produce vaccine and to reach all people in need of vaccine. So one government can also fund the companies that are producing vaccine so that more number of people, those who are in need of vaccine, can be benefited out of it. Yeah. Second question. So glucose is used in the synthesis of amylose. Glucose is first converted to glucose 1 phosphate, that is G1P, and starch phosphorylase is an intracellular enzyme that can catalyze the synthesis of amylose from G1P, which is the substrate for this reaction. So here glucose 1 phosphate is getting converted to amylose and some phosphate ions are being released. So student used calorimeter to investigate the progress of this reaction. So the student made a reaction mixture containing 0.01 mole decimeter cube of G1P in a buffer solution at pH 6. A very small quantity of amylose was added to initiate the reaction. So this amylose was added just to start the reaction. Then a solution of starch phosphorylase was added to the reaction mixture, and samples were taken at 1 minute intervals. Each sample was added to a dilute iodine solution, stirred and then poured into a cuvette. The absorbance of each solution was recorded. So here the results for this reaction is shown in the form of graph, where the x-axis shows the time after addition of starch phosphorylase per minute, then absorbance is shown on the y-axis. So what is the question? So A part, explain why the absorbance increases as shown in figure 2.1. So as you have seen in the previous slide, in the graph the absorbance value increases. When the time increases, the absorbance also increases. So we just need to explain why the absorbance increases. So amylose react with iodine to give blue black color. So as the reaction proceeds, more amylose will be produced with the help of the enzyme starch phosphorylase. So this amylose will going to react with iodine to give deep blue black color. Then second part of A question, a student took their final sample at 12 minutes. Predict the results for absorbance if the students had continued to take samples for further 10 minutes, and explain your answer. So here we are just going to predict what would be the result if the students are going to continue the experiment for further 10 minutes, and for our prediction we need to give a proper explanation. So here as we see in the graph, like the absorbance value is increasing as the time increases, but the prediction what could come here is like absorbance reaches constant at 1.80. Why this absorbance reaches constant at 1.80 is because the maximum number of value what we can get through the absorbance is 2. So beyond 2 the absorbance will not increase. So at 1.8 itself it should reach constant, or if not, the absorbance may increase very slowly after 1.80 till the value 2. So the proper explanation for this is the substrate G1P might be used. So because of this could have been happened, or substrate concentration is a limiting factor, then calorimeter reaches its maximum absorbance limit of 2.0. Third part, state an advantage of using a calorimeter in determining the progress of the reaction. So what is the advantage of using calorimeter? So why can't we just look through the color, look the color with our naked eye. So what is the advantage of using colorimeter here, they are asking. So colorimeter is going to give us qualitative data that can be used for plotting graph. So colorimeter gives a qualitative data where the digital reading can be seen, which you can use it for plotting the graph. Then color is not judged with eye. So we are not judging the color with naked eye, stating that yes, this color, one set of test tubes or one set of cuvettes are having light blue color and the other set of cuvettes are having deep blue color. So we are not judging with eye, so it is being judged with the help of colorimeter. So this is an overall advantage of using colorimeter in determining the progress of the reaction. B part of this question. So the reaction catalyzed by starch phosphorylase occurs at the end of amylose molecules. Describe the sequence of events that occurs when starch phosphorylase catalyzes the addition of a molecule of glucose to the end of an amylose molecule. So here we have to write only about the sequence of process, how enzyme and substrate is going to react. So the basic question is the second part of this question. We describe the sequence of events that occur when starch phosphorylase catalyze. So this is the question what we want to understand here. So what happens when the substrate and enzyme is going to react together? So that is the major question asked for 4 marks here. So what are the process will happen when an enzyme and substrate is going to react together is the substrate glucose or the G1P will bind to the active site of starch phosphorylase to form enzyme substrate complex. So end of amylose bind to the active site. As the amylose act amylose act as the substrate, so it binds to the active site. So amylose only bind to the active site of the enzyme, and active site changes shape when substrate is bind to an enzyme. So this goes with the induced fit hypothesis. Then enzyme decreases the activation energy as the enzymes are present here, so the activation energy is being lowered. Third question, hemoglobin is a complex protein molecule made of 4 separate subunits as shown in figure 3.1. So hemoglobin is a third question. Hemoglobin is a complex protein molecule made of 4 separate subunits as shown in figure 3.1. So the structure of hemoglobin is given in the diagram. So here the question asked here is the shading represents 2 different polypeptides that form a molecule of hemoglobin. So what are the 2 different shadings? So this might be in black and white color for your understanding. It is being shown in the color. So you have 2 types of polypeptides that are present in the hemoglobin, and the name of the 2 different polypeptides are being asked. So hemoglobins are formed by 2 polypeptides, mainly by 2 polypeptides. One is the alpha globin and the other one is beta globin. And identify the structure labeled P. So the structure labeled P is nothing but hem group or the prosthetic group, which is made of iron. Then figure 3.1 shows some of the levels of protein structure. State the level of protein structure that is not shown. So 3.1, the image what is given here, which is 3.1, so it shows the level of protein structure, and they are asking which level of structure is not shown in this. So as we all know, the hemoglobin is the quaternary protein, quaternary globular protein, which is formed by different tertiary protein. So here the structure which is not shown here is the primary structure. The basic amino acid sequence is not shown here. So the basic structure, the basic amino acid structure or the primary structure of amino acid is not shown here. Moving on to B part of this question states, hemoglobin is involved in the transport of carbon dioxide. Molecules of carbon dioxide enter the red blood cells as the cells travels in the capillaries through muscle tissue. Some of these molecules are converted to carbonic acid. Explain how hemoglobin is involved in the transport of carbon dioxide molecules that are not converted to carbonic acid. So most of the carbon dioxide are being converted to carbonic acid and they travel through the blood plasma to the heart. But what is how the carbon dioxide molecule that are not converted to carbonic acid, how they are going to be? B part of this question asks, hemoglobin is involved in the transport of carbon dioxide. Molecules of carbon dioxide enter red blood cells as the cells travel in capillaries through muscle tissue. Some of these molecules are converted to carbonic acid. And the question asked here is, explain how hemoglobin is involved in the transport of carbon dioxide molecules that are not converted to carbonic acid. So some of the carbon dioxide will be converted to carbonic acid, but how the carbon dioxide which are not converted to carbonic acid are transported with the help of hemoglobin is the question being asked. So here carbon dioxide molecule, first they will be combining with hemoglobin. So where do they go and combine? So they combine at the terminal amine group of hemoglobin and they are going to form carbaminohemoglobin, and carbon dioxide will be combined with hemoglobin till the pulmonary circulation. So pulmonary circulation here, like when they are once they formed into carbaminohemoglobin, they will be traveling along with the blood vessels, and they till they reach the lungs in the alveoli till the pulmonary circulation ends. So they till the exchange of gases, the carbon dioxide and oxygen exchange of gas, till that time the carbon dioxide will be combined with hemoglobin in the form of carbaminohemoglobin. So this is the answer. C part of this question: when hemoglobin associates with oxygen, it forms oxyhemoglobin. State the precise site in the mammalian body where hemoglobin molecules bind with oxygen. So it is a very direct question. We are just need to answer where hemoglobin will be binding in the. C part of this question asks, when hemoglobin associates with oxygen, it forms oxyhemoglobin. State the precise site in the mammalian body where hemoglobin molecules bind with the oxygen. So where does hemoglobin molecule bind with the oxygen? So it is very simple, like oxygen is going to combine with hemoglobin with in the capillaries that are found around the lungs. So inside the alveoli, like from the alveoli, the oxygen will get diffused to the capillaries. So there oxygen will be combining to the hemoglobin molecules. Moving on to D part of this question, the compound 2,3-diphosphoglycerate 2,3-DPG is produced in red blood cells. 2,3-DPG binds to hemoglobin and stabilizes it. Figure 3.2 shows oxygen dissociation curves when red blood cells have high and low concentration of 2,3-DPG. So here the curve that is on the right hand side shows the high concentration of 2,3-DPG, and curve that is on the left shows the low concentration of 2,3-DPG, and x-axis shows the partial pressure of oxygen kPa, and y-axis shows the percentage saturation of hemoglobin with oxygen. So questions. P50 is the partial pressure of oxygen when hemoglobin is 50% saturated. The P50 is used to compare the affinity of hemoglobin for oxygen under different conditions. Use the information in figure 3.2 to describe the effect of an increase in the concentration of 2,3-DPG on the oxygen dissociation curve. So the question here asked is very simple. Without any confusion, we can just look into the graph. So they are just asking, use the information in figure 3.2. So this is a graph which we have seen in the previous slide. So in figure 3.2, to describe the effect of an increase in the concentration of 2,3-DPG on the oxygen dissociation curve. So when there is a high concentration of 2,3-DPG, what are the effects that are going to take place, they are asking. So what are the effects? So from the graph, it is very clear that the concentration of 2,3-DPG, the graph shifts to right. Then percentage saturation of hemoglobin with oxygen decreases. So the percentage saturation of hemoglobin is going to decrease when the concentration of 2,3-DPG increases. The percentage saturation of hemoglobin with oxygen decreases. More partial pressure of oxygen is required to reach the same percentage saturation as low DPG. Second part of this question, blood is stored in blood banks for use in hospitals during operations. The concentration of 2,3-DPG in red blood cells decreases when blood is stored in blood banks. State and explain the effect that the use of blood taken from a blood bank has on the supply of oxygen to the tissues of a person during an operation. So it is very simple. They are asking what is the effect of blood taken from the blood bank in supplying oxygen to the tissues of person during an operation. So hemoglobin, initially the hemoglobin will takes up all the oxygen and tissue has less supply of oxygen. Moving on to fourth question. So the fourth question, figure 4.1 is a diagram showing the transcription of part of the COL1A2 gene that codes for collagen polypeptide. The part of the COL1A2 gene shown is a section of exon. Structure A represents an enzyme involved in transcription. So the first question, A1, name the enzyme labeled A in figure 4.1. So before moving on to this question, we should know that they are talking about transcription. So transcription involves the synthesis of mRNA. So the enzyme which involves in the synthesis of mRNA is RNA polymerase. So DNA polymerase will be coming into play during the DNA replication, but here it is the synthesis of RNA. So RNA polymerase is the enzyme which is being used for the synthesis of mRNA. Then name the bond that forms between the nucleotides in the primary transcript. Primary transcript is nothing but the mRNA. So what is the bond that formed between the nucleotides? So all the nucleotides are going to be bonded together by phosphodiester bonds. Then state the number of amino acids that are coded for by the sequence of nucleotides on the primary transcript shown in figure 4.1. So they are asking number of amino acids that are coded by this primary transcript from the figure. So we know that amino acids, like one triplet code forms one amino acid, so we should count how many triplet codes are there. So to count that, I have just made some markings there. So this CUU, UAC, AAG, AGG, UGU is the there are five triplet codons which are present in this primary transcript. So 5 amino acids are being produced or coded by this primary transcript. Then second part, use the information in figure 4.1 to explain why one of the strands of DNA is not transcribed. So from the figure we can able to see that only one strand is being used as for transcription. The other strand is not used, and they are mentioned as non-transcribed strand. So what is the reason for this? So only the template strand will be transcribed, other strand will not be transcribed. So if the other strand is transcribed, may result in a different amino acid sequence which might synthesis a non-functional protein, or in case when you are doing this, a lot of energy is being unnecessarily used. So just to avoid this, only one template strand will be there which is going to code for the amino acid which is going to code for the amino acid which need to be synthesized, or the polypeptide that need to be synthesized. So RNA polymerase will only synthesis mRNA from 5 prime to 3 prime direction. So 5 prime to 3 prime direction will be possible only in one direction as a leading strand, from 3 to 5 direction. If the transcript strand is in 3 to 5 direction, the primary transcript produced will be in 5 to 3 prime direction. C part of this question, new mitochondria are formed when a mitochondria divides into two. So figure 4.2 is a transmission electron micrograph of mitochondria that is dividing. So the question asked here is, state two features of mitochondria that are visible in figure 4.2. So first one, like cristae. Cristae is very well visible in the mitochondria. And second one, mitochondrial matrix. Double membrane and like inner and outer membrane can also be stated, but more clearly visible structure is the inner cristae and mitochondrial matrix. Even 70S ribosome can also be written for this answer. Second part of this question, the line on figure 4.2 shows the diameter of one of the mitochondria. Calculate the actual diameter of the mitochondrion, and they are asking you to give the answer in one significant figure, and the magnification of the mitochondria is given as 50,000 times it has been magnified. So we all know the formula to find the actual size, like A is equal to I by M. So measure the length in centimeter or millimeter and divide it by the magnification. So here the length is 3 centimeter, which when we convert this into millimeter, which becomes 30 millimeter, and 30 divided by 50,000 gives us 0.0006. So if you are going to convert that into micrometer, since it's asked in micrometer, so 0.0006 into 1000, which is 0.6. So the answer is 0.6 micrometer. Yeah. Moving on to next question. Mitochondrial DNA codes for some polypeptides of protein used within the mitochondria. Some of the proteins allow movement of ions into and out of the mitochondria. Outline the ways in which ions can move into mitochondria. So here the question directly asked here is how ions are going to pass through the cell membrane. So what are the ways the ions can take to pass through the cell membrane? So first, ions move by active transport against the concentration gradient using ATP. It's the first way. Then second, ions can also move by facilitated diffusion. Then protein carriers or protein pumps undergo conformational change and carry the ions. So for all these things to happen, like active transport, all those things, the protein carriers or protein pumps undergoes conformation change and carry the ions inside the cell. Yeah, moving on to next question. In flowering plants, transport of assimilates occurs in phloem tissue between sources and sinks. Explain why a root can be a source and a sink. So here they are asking us to explain why the root can act as both source and the sink. So when plant is germinating, root provides assimilates for other parts of the plant where the root is going to act as a source, and at the same time assimilates from leaf will also be stored in root where the root is going to act as a sink. So apart from this, phloem is also bidirectional, so it can move in both the directions. Then second part of this question, state two examples of assimilates that are transported in the phloem. It's a direct question where we can simply write sucrose and amino acids are the assimilates that are being transported in the phloem. B part of this question, describe and explain the mechanism that is responsible for the movement of phloem sap in sieve tubes. So, movement of phloem sap in sieve tubes. So, initially we should know that companion cells, phloem sieve tube cells are will be together, and through plasmodesmata, things are going to move from companion cell to phloem sieve tube. So, what will happen here? Assimilates from companion cell diffuse into phloem sieve tube, and this decreases the water potential of the sieve tube element. So water from the nearby cell enter inside the sieve tube element as a result of osmosis down the water potential gradient. This develop the buildup of hydrostatic pressure in the phloem sieve tube at source. Hydrostatic pressure will be high at source. So source is the place near to the companion cell where the water is getting inside through the process of osmosis. Because of this, mass flow happens because of the pressure difference. Then assimilates move to sink followed by the water. Moving on to next question. Tobacco mosaic virus (TMV) infects many crop plants. The virus passes between cells in the leaves and can travel throughout the plants in the phloem. The enzyme pectin methylesterase (PME) is involved in the production of cell walls. The enzyme is also known to influence the movement of TMV through plants. Scientists investigated the effect of PME on the transport of TMV through plants. The scientists used three varieties of tobacco plants. Two varieties, V1 and V2, have small quantities of PME, and a third variety, C, has a normal quantity of PME and was used as a control in this investigation. The plants in each group were infected with TMV at the same time. The accumulation of the virus particles transported to the leaf at the top of the plant was determined over 36 days. The results are shown in figure 5.1. The arrow indicates when all the plants were infected with TMV. So here, so the question asked here is compare the results for varieties V1 and V2 with the control group of plant C. So accumulation of TMV is delayed in both the varieties V1 and V2 when compared to C. So till day 9 there is no increase, and from day 16 there is a steep increase in the variety V1. Then again both variety V1 and V2 also see all three reach the maximum level of infection. Then small quantity of PME does not decrease as the maximum accumulation of TMV. Similarly, normal PME is not needed to have a maximum accumulation. That is, when we reduce the level of PME, the maximum accumulation is not decreased. So it has reached the maximum level of accumulation. Similarly, when normal PME is not needed to have the maximum accumulation, that is, even if the small quantity of PME is there also, the maximum level of accumulation is seen. Question 6. The mammalian heart consists of 4 chambers, and A part, explain why the ventricles have thicker walls than atria. So we all know that usually ventricles will have thick walls when compared to the atria. So the reason for this is, like, ventricles, they are going to pump blood to a greater distance, like pulmonary and systemic circulation. Then ventricles have more cardiac muscle than atria to pump blood with higher pressure, and they do this to overcome the high resistance. Here in this question we have just compared both left and right ventricles together, and we have not differentiated that left ventricle is going to pump blood with great pressure when compared to the right ventricles. Here the question asked is commonly about ventricles have thicker walls, so we should state in common why ventricles, both right and left ventricles, they are going to have thick walls when compared to atria. So this answer goes for this. B part of this question, complete the table 6.1 to identify the structure in the heart responsible for each function listed. So the table 6.1 is given with some list of function and structure in heart, that which is going to do that function we are going to identify. So initiate the heartbeat. So heartbeat is initiated by the sinoatrial node, that is SAN, SAN. Then delays the wave of depolarization between the atria and the ventricle is the atrioventricular node, that is AVN. Then transmit the wave of depolarization through muscles of the ventricles, which is Purkinje or Purkinje fibers. Then closes when the left ventricle contracts. So when left ventricle is going to contract, which is going to close? So it is the bicuspid valve or the mitral valve. So that's it with this paper. I hope you would have understand the concepts and the explanation. Please do like, share and comment your thoughts and subscribe for my channel. Thank you.